I'm gonna pop some carbs. I got no glucose in my pathway. I'm busy looking for some cold. This is freaking awesome.
Nah, I got some glucose, hell yeah, I'm in that pep phase. I'm so pumped doing work with hexokinase. Glucose to the glucose with the number 6 phosphate. PGI to that fructose 6 phosphate. ATP, ADP, phosphofructokinase E. 1,6-bis, FEP, autolase to GAP. GAPDH with some PGK and PGN. PP to the 2-phosphoglycerate and then PEP to pyruvate. FAD, NAD, about to go and grab some hydrogen. Pumping out all the substance for some work, yeah, that's the energy. Shuttling through that mitoc like nobody's business. What you know about oxidation, bitch? I'ma do with TCA, I'ma do with TCA. No, for real, moving on. Can I have some AKA? Citrus synthase from lactoate to that citrate. 8 kinase, step 2, isocitrate. I had some NAD+, I used some NAD+. I made some NADH and avocado glutamate. Dehydrogenase complex, put some CoA. CO2, NADH out, sucks my CoA. Then I take that GDP out of P. Hold on, wait, always gone. People be like, ah, he got the succinate.
I'm gonna pop some carbs. I got no glucose in my pathway. I'm busy looking for some cold. This is freaking awesome. I'm gonna pop some carbs. I got no glucose in my pathway. I'm busy looking for some cold. This is freaking awesome.
What you know about intermediate metabolism? What you know about what's going on in your skin? I'm digging, I'm looking for some FAD. Step 6 succinate dehydrogenase, that's the key. Moving on, reducing that fat up to FADH2 fumarate. That's the enzyme that goes strictly based to malate. Step it, malate dehydrogenase. Finish off NADH oxaloacetate.
I'm gonna pop some carbs. I got no glucose in my pathway. I'm busy looking for some cold. This is freaking awesome. I'm gonna pop some carbs. I got no glucose in my pathway. I'm busy looking for some cold. This is freaking awesome. That's how Krebs cycle goes. If you didn't know, now you know. This is how TCA flows. BCH actually doesn't know. That's how Krebs cycle goes. If you didn't know, now you know. This is how TCA flows. BCH actually doesn't know. I'm gonna pop some carbs. I got no glucose in my pathway. I'm busy looking for some cold. This is freaking awesome.
I forget a little.
What a better way to start a lecture on the Krebs cycle, the TCA cycle, the tricarboxylic acid cycle, the citric acid cycle. They're all the same thing. They're all referring to what do we do with the acetyl-CoA that's made by the pyruvate dehydrogenase complex. And just in case you didn't know, when they said BCH doesn't blow, that actually, BCH stands for biochemistry, just in case you were wondering. So, with this here is kind of a schematic of the citric acid cycle. Essentially what happens is you're going to get some oxaloacetate, which is a 4-carbon receptor. It picks up the 2 carbons that comes from the acetyl-CoA that was made, and that then becomes a 6-membered 6-carbon species. It begins with citrate, then it moves its way through. Then you get a couple of decarboxylic acids, decarboxylations, so you spit out some CO2, and these CO2 molecules, it turns out, were CO2 molecules that were already present on this C4. They weren't these two here. They're actually, as it goes around the cycle, what you'll find out is that the CO2 carbons, the carbon dioxides that are coming off are actually coming off of this oxaloacetate, and essentially these guys shuffle down, and eventually through enough rounds they will also eventually be the carbons that come off as carbon dioxide. You also get NADH made here, NADH, FADH. So we get 3 NADHs and 3 FADH2s. I mean, sorry, 1 FADH2. That's just for one round of the Krebs cycle. Remember, there's going to be 2 rounds of the Krebs cycle for every glucose, because we split the glucose into 2 3-carbon molecules. So each of those 2 3-carbon molecules became pyruvate. Each one of those pyruvates from the initial, each one of the 2 pyruvates that came from glucose then was created 2 acetyl-CoAs. And so this is just one part of the one round represents half of the glucose, the other round is the other half. So we get 1, 2, 3 NADHs from one round, and FADH we get as well, and we'll find out later where these come in. We actually will cache these in. Embedded in the H here is extra electrons. It's called a hydride was transferred, and there's extra electrons there, and these electrons get passed into something called the electron transport chain, which helps to generate a proton gradient, and then that proton gradient is later harnessed. The energy that gets stored in the proton gradient is harnessed to make ATP. So that's essentially what's going to happen there. Now this ATP molecule that gets made actually comes from GTP that gets made, and then that can be used to create ATP. So that's what we're going to be talking about. The Krebs cycle, or the TCA cycle. Now, kind of looking at this, let's look at the things that feed into the Krebs cycle. Well, or the citric acid cycle. First of all, glucose, and that's the main pathway that we've talked about. But we also just, if you recall, I showed you the little video that had to do with pyruvate dehydrogenase complex deficiency, and that young lad who had that couldn't actually use glucose. It caused him problems because every time he ate glucose, the block to get into the citric acid cycle through pyruvate dehydrogenase complex was in place, so he had that deficiency. So he had to go through anaerobic respiration in that case, and that caused the lactic acid buildup. Well, they said they gave him a ketogenic diet, which is fatty acids and amino acids that come from protein. So he had a high fat, high protein diet. And that's because the fatty acids and the amino acids can be converted also into acetyl-CoA and enter into the Krebs cycle without needing the glucose. It brings its own challenges, obviously, as you might guess, given what we know about fatty diets and their impact on health. But the fact is that kind of a modified diet was enough to bypass the blockage in glucose metabolism for that person. But anyway, the bottom line is these different routes to get to acetyl-CoA then are feed-ins to the Krebs cycle. And then, as I mentioned before, these 8 electrons actually come from these. You get 2 electrons per NADH and 1 per FADH. So those electrons get fed into the electron transport chain, and water is made. And that's what we see here. Oxygen is the final acceptor of the electron transport chain, and the very exergonic reaction that results is the formation of water. So that drives all this process forward, because all of these, no matter how favorable they are linked to a very, very favorable reaction, that is the formation of water, and that's enough to pull all of these reactions forward, because this is so favorable. And then, as one of the byproducts of going through the electron transport chain, is the generation of this proton gradient, and the proton gradient then is harnessed through an enzyme called ATP synthase, which essentially uses the proton gradient to fuel the addition of the phosphate onto ATP. So you can see the connection. This is kind of the big picture where we're going to be heading in the next 2 weeks. But we're going to start right here with the acetyl-CoA and the Krebs cycle. So just like we could separate the the glycolysis into 2 stages, we can also do the same with the Krebs cycle. So the Krebs cycle can also be fed into 2 stages. The first stage generates 2 molecules of carbon dioxide by a process known as oxidative decarboxylation. That's the fancy word. So here's an example of what's happening. Let's go over some of the steps in the first stage, the stage that leads to oxidative decarboxylation. So the very first reaction, the opening gambit, so to speak, is oxaloacetate, which is a 4-carbon acceptor unit here, picks off the 2 carbons that are attached to acetyl-CoA, and citrate synthase does this reaction, and it creates citral-CoA as a brief intermediate, and then you get hydrolysis of the CoA, and essentially hydrolysis looks like this. You have your water molecule here, and it attacks here, and that leaves. That's hydrolysis, essentially. And one of these, from a stoichiometric point of view, one of these protons gets transferred to the CoA to form CoAsh. This and this are the same. This just means this is CoA with the free sulfhydryl, is usually how I've always written it, but textbooks oftentimes will say CoA. I've studied this enzyme actually as a grad student because it was a enzymatic cousin to 3-hydroxy-3-methylgluteryl coenzyme synthase that I studied, but it used similar kinds of chemistry. So this reaction now kind of primes the cycle. Now it's ready to go. So acetyl-CoA, this is reaction 1. Acetyl-CoA enters the cycle as it gets attached to this oxaloacetate, and that leaves us with citrate. And you can see these magenta colored carbons are to remind you which carbons were added into this cycle. These carbons that are present here, these 4, came from previous cycles. So every time 2 carbons enter in, 2 carbons leave. 2 carbons enter in, 2 carbons leave. And so that's how this works. So that's where those oxidative decarboxylations take place. That the carbons leave as carbon dioxide. So let's see what that looks like. But before we do, let's talk a little bit more about the reaction mechanism of citrate synthase itself. Citrate synthase uses induced fit. If you recall what induced fit means, that it means that there's a conformational change that brings oxaloacetate causes this conformational change that induces structural changes that lead to the new binding site that is now recognized, is recognized by acetyl-CoA. So first oxaloacetate binds, so it's a 2-substrate reaction. First oxaloacetate binds, and when it does, that creates a change in the site that now it creates a new site that recognizes acetyl-CoA. This is kind of a theme you'll see in 2-substrate reactions. A lot of times what will happen is when the first substrate binds, it creates a second substrate binding site where then that molecule, whatever that second substrate is, can bind. Now that's not always the case, but in the case of citrate synthase, it is the case. So this kind of depicts that. You can kind of see where there's some open binding sites where the oxaloacetate can bind, and when oxaloacetate binds, you get these sites clamped down and create new sites for the acetyl-CoA to bind. So that conformational change opens up new sites that recognize acetyl-CoA. So one thing before I go any further, I want to make sure you understand that the Krebs cycle happens in the mitochondria. So glycolysis happens in the cytosol, and then the pyruvate that gets into the mitochondria and gets converted to acetyl-CoA in the mitochondria, and then Krebs cycle happens in the mitochondria. So all this is happening in the mitochondria. I'm mentioning this because of this enzyme. It actually has a dual role in the cell. So I will first remind you, go over what it does when it's in the mitochondria. Then I'm going to mention to you what this enzyme can do in its dual role, which I find really interesting because they just started, when I was in graduate school some years ago, they actually they just discovered this other role for this enzyme, and it was kind of the hot topic while I was in grad school. So it was historical, which I guess means I'm old. Anyway, so we get citrate synthase, takes oxaloacetate, combines it with acetyl-CoA to form citrate. Then aconitase comes in. Aconitase is an interesting enzyme. It actually contains something called an iron sulfur cluster. And I've mentioned this because aconitase uses this iron sulfur cluster in order to facilitate the chemistry that you're seeing here. In this chemistry, you get the conversion of citrate to isocitrate. So you can kind of see that this is in effect, it involves reduction and oxidation. And the iron sulfur cluster is a tool that enzymes often use to do this kind of thing. And the iron sulfur cluster is there's all different types of iron sulfur clusters, but they're ways that proteins, by binding to the iron sulfur clusters, it extends the kinds of chemistry that can be done that goes beyond what can be done by amino acids alone. So this is an important part. And for that other thing that I told you about, that the other role that aconitase plays in the cell, it also uses its iron sulfur cluster, but it doesn't use it to do oxidation and reduction chemistry, and I'll tell you what it does in a minute. So the bottom line is you get this formation of citrate to isocitrate, and this enzyme's interesting because I learned a lot about it in grad school, because two of the kind of well-known biochemists in the world, actually, who are studying aconitase worked at the medical college. One of them was his name is Helmut Beinert, which he was very old back when I was a grad student, so I'm certain he's passed away. But he's done many seminal biochemical experiments, and he pioneered the use of all kinds of techniques that we currently use regularly these days, and it's traced back to him. So it was a great honor to be taught by him. And then he also had another colleague that worked in his lab, and her name was Claire Kennedy, Mary Claire Kennedy. And what's interesting about her is she was a nun who was a scientist, and she helped me a lot in understanding chemistry. Actually, she was a great person. I often had to consult with her to learn about my own project and the kinds of things I was doing. But anyway, one of the things I remember learning about from them was this poison called fluoroacetate, sorry. And fluoroacetate turns out to be a substrate for aconitase. Essentially what can happen is fluoroacetate gets converted to fluorocitrate, and fluorocitrate then can bind with such high affinity to aconitase that it can just shut it down and stop it from working. So in this way it's called a suicide inhibitor, because actually aconitase can convert fluorocitrate through a series of steps. Fluoroacetate gets added and you get fluorocitrate. Anyway, the bottom line is aconitase converts this into the inhibitor that can kill it. And so it's kind of a really interesting side reaction that it can create this fluorocitrate that then leads to death of the enzyme. And obviously this would be a pretty potent poison for the organism as well, because if you don't have aconitase working, you're in a world of hurt. So anyway, back to the other job of aconitase in the cell. Because of this iron sulfur cluster, when aconitase is found in the cytosol, so when aconitase is found in the cytosol, it's the same exact protein. Here's a deal where people purified a protein in the cytosol that actually they named it iron regulatory binding protein and iron regulatory element binding protein. So that's what they came up with. And when they sequenced it and found out what it was, it had the same exact amino acid sequence as aconitase. And it turned out that this iron sulfur cluster, when there was low iron in the cell, it would not have all the irons present that it would normally have in a happy iron sulfur cluster, and it would then serve as an iron sensor in the cytosol. So, and interestingly, this iron regulatory element binding protein, I think it's called IRBP1. It is fully functional in running this reaction. So when it has its full iron sulfur cluster in the cytosol, it still acts as aconitase. It can still do that reaction. So I just find that very fascinating. So here's a case where an enzyme is actually serving two very different roles, but use the same functional group to do both. And so that's really, I find, intriguing. And I just remember that kind of discovery. They were like shocked. Wait a minute, you mean that iron regulatory element binding protein is aconitase? And I just remember that kind of caught everyone off guard, because it actually introduces a really interesting model for how you can get diversity out of your gene products by just where they are found. When they're in this case, in the cytosol, has a totally different role than when it's in the mitochondria. Yet it's fully able to do the reaction regardless of where you find it. That's just really cool to me. So in the next reaction, we take the isocitrate that's been made, and using NAD, NADH is created, and that NADH then is that process, NAD, will cause oxidation to happen at this carbon. You see that? You get a carbonyl there. And you get this intermediate known as oxalosuccinate. And then you get acid, an acid catalysis here, general acid catalysis, where a proton is added. And this that gets joined up here, and essentially you get a decarboxylation, and the electron that's left behind on the carbon gets protonated. That's essentially what's happening. And here's just something I've noticed whenever you get, this is a beta carboxyl group. It's beta to a carbonyl. Whenever you get a beta carboxyl group to a carbonyl, it's pretty unstable and quite readily able to be lost in the decarboxylation reaction, and that's exactly what's happening here. So the strategy that this enzyme uses is it uses oxidation to create this pretty happy to be dumped carboxyl group then, and that part of that has to do with this thing can what can happen is you can get these electrons going here, and you can get the keto-enol sort of resonance structure, and that's what helps this to bump off, because you get extra electrons here from the double bond. So at any rate, then you get this alpha ketoglutarate. So NADH is released, and this NADH can now go in and be cashed in for ATP down the road, and we'll talk about that. And when we talk about electron transport and oxidative phosphorylation. So this is the first of two oxidative decarboxylations that take place. So one carbon's lost in this process. Then alpha ketoglutarate gets converted to succinyl-CoA, and this also represents another oxidative decarboxylation. So in this case, we go from NAD to NADH, NAD to NADH, and the coenzyme A is utilized to the coenzyme A is attached, and this has a very similar set of reactions to pyruvate dehydrogenase, if you remember, where you actually get an attachment. In this case, you'll get because this would be, instead of pyruvate, now you have alpha ketoglutarate. This carboxyl group is actually pretty happy to leave as well. So if you have some kind of a sulfhydryl attack, just like we had with the lipoamide and all that, if you remember, eventually we got acetylation, and then CoA would displace it. We also had oxidation take place, and to help convert this into something like a carboxyl group, because then you get this thioester, right? So the thioester is really an activated carboxyl group in the end. At any rate, you form succinyl-CoA and NADH, and you get another decarboxylation. So the NADHs, we just created two NADHs, and NADHs in the Krebs cycle mean ATP. And what you'll find is for every NADH, you'll create, down the road, we'll see this, 3 ATP for every NADH that we cache into the electron transport chain. Welcome to stage 2 of the Krebs cycle. Here's where we began to harvest and grab some energy. And the first energy step that we get here is we get the formation of GTP. And GTP actually can then be used. So GTP to GDP can be used to transfer the phosphate from GTP to ADP to form ATP. So you can get this, that's what this is saying here. So here's where we're converting thioester energy into the formation of what we call a substrate level phosphorylation reaction. Kind of reminds me a little bit of glyceraldehyde 3-phosphate dehydrogenase, where we actually involve inorganic phosphate getting to a point where it can phosphorylate directly. So we're harnessing the energy of this thioester to put a phosphate onto GDP. So succinyl-CoA synthetase creates succinate from succinyl-CoA and also GTP. So that's where we can get directly right from the Krebs cycle without having to cash in the NADHs and FADH2s to get ATP. We can get one ATP just from this reaction alone. And remember, every glucose means 2 rounds through the Krebs cycle. So what this actually means is you would get, for every glucose, 2 ATP out of this step. So in that way, succinyl-CoA is a way to convert the thioester into ATP, which through a series of steps via GTP formation. So here is the reaction. As you can kind of see it, you get this histidine helps orient the phosphate for a nucleophilic attack on this high energy succinyl-CoA that leaves the CoA as a leaving group, which is a very stable leaving group. That's why it works. Then you form this succinyl phosphate, and so that's kind of interesting because really we're using this high energy thioester to create another high energy phosphorylation step here. Then you get a transfer of the phosphate to the enzyme itself, so you actually get a histidine, a phosphohistidine formed, and that's why we call it substrate level phosphorylation, because the substrate actually helped the phosphorylation process. And then ADP comes along, and ADP can the phosphate, the beta phosphoryl oxygen can attack this phosphorus center, and that will allow this to leave, and you get ATP formed. In the slide just before it, we also mentioned GDP as well and GTP. Well, it turns out that both of these can take place. It just depends on the enzyme. There's GDP favored or GDP favored succinyl-CoA synthetase enzymes, and actually in that way it can run backwards. A lot of times what happens is GTP gets converted to GDP to form succinyl-CoA, and that succinyl-CoA can, so that takes succinate, and it can create succinyl-CoA by running the reaction backwards. A lot of times the enzyme that does that backwards uses GDP, and it can go both directions, but a lot of times GTP is used for the backward reaction, because this actually is a precursor that gets used to make other things, one of them being heme. You remember the heme groups in hemoglobin. So there are enzymes that can run this with ADP and ATP. You can also run this with GDP going the GTP, and this GTP can be converted to ATP, like I said before, or sometimes this reaction runs backwards when we need to make a metabolic precursor to make a molecule. So when we have plenty of energy, then as we'll find out, that the intermediates from the Krebs cycle, when there's plenty of energy, can actually be siphoned away and used to make other molecules. Molecules you need for other purposes. Anyway, bottom line is it's complex, but you get the idea. I just didn't want you to get confused by saying, wait, you just talked about GDP and now you're bringing up ATP. Well, both of them work. In the way it's mentioned in your textbook, it says that it utilizes a purine, adenosine, or guanine are purines, so it uses a purine diphosphate. So at any rate, in the E. coli enzyme, it happily uses either of those. And then when in eukaryotes and other things, typically it's ADP, except in the case that I just mentioned when they work backwards. And a lot of times it uses GTP and works backwards. So this enzyme, all these reversible arrows mean that this can all happen both directions. And so it all depends on whether or not the cell needs energy, or it has enough energy and can do other things with the intermediates from the Krebs cycle. And then we can sort of create oxaloacetate again, and that uses a couple of enzymes, 3 of them. Successively, we get succinate dehydrogenase. So that takes succinate to fumarate, and that generates FADH2. And FADH2 can go on into the electron transport chain and be cashed in for 2 ATPs, it turns out, as we'll learn. So that can be cashed in for 2 ATPs. And it's not exactly 2. It'll be like, I forget what it is, something like 1.9. It generates enough energy to do that, so we always round it. And typically in textbooks, that's often what you'll see. They just round it to the nearest ATP. So you get 2 ATPs from that. And then in the next reaction is fumarase. You go from fumarate to malate, and you can see that it's adding water across that double bond. So it's hydrating that bond. So that's what the reaction, so that's what fumarase does. And then malate dehydrogenase, listen, every time you hear the word dehydrogenase, it's a good indication that that step is going to involve either the production of FADH2 or NADH. Dehydrogenases tend to either use or create NADH. Just think of it that way. In this reaction, it creates NADH, and that again, that's malate dehydrogenase, takes malate and oxidizes. That's what it's doing, is oxidizing that carbon to form this ketone here, and now you get oxaloacetate. Guess what? You've just completed the cycle, and now another molecule of acetyl-CoA can come in and do the whole thing all over. So the energy-rich groups, you can kind of keep track now what happened as we've gotten electrons moved around. Remember, loss of electrons is oxidation, gain of electrons is reduction, so we can kind of see what's happening here. The carbons are being oxidized. They're losing electrons, and what are they losing them to? Well, they're losing them to NAD and FAD to form NADH and FADH. So this is getting oxidized, these are getting reduced. Okay? And then we get the formation of ATP, and we get protons. This goes back to what I was saying. I said 2.8, it's 1.8. So essentially it's 2.5 ATPs and 1.5 ATP for 2.5 for NADH, 1.5 for FADH2. A lot of times you'll see 3 and 2. because they round to the nearest molecule, but it's more like when you calculate the energy that you get from the whole thing, it's more like 2.5 ATPs and 1.5 ATP. So here it is. Here's the whole thing put together. So you get the entry point here is right here. So here's reaction 1, 2, 3, 4, 5, 6, 7, 8. So there's 8 reactions in the Krebs cycle. Citrate synthase forming citrate. Then citrate gets converted to isocitrate using aconitase. Then you get a series of 2 back-to-back oxidative decarboxylation reactions. So you get 2 CO2s given off, and then you begin your energy. Kind of, this is the second phase here, and you create ATP. You get FADH2 and NADH as you work your way through regenerating oxaloacetate, and then the next molecule of acetyl comes in, and it just does it all over again. So that's the cycle form of it, but I wanted you to see this. I'm particularly interested in this reaction mechanism, succinyl-CoA synthase. I really want you to know that reaction mechanism and know it well, because I find it really interesting. There's a lot going on there, and the chemist in me can't pass this up without making you better understand it. And since the book outlines it too, that'll be my compromise, because I really like going through. I love this pathway, this one too, but I'm just going to make you know this one. You have to know this reaction mechanism. I want to make sure you understand what I mean by that. I mean, I want you to know the chemistry of the steps here. I do want you to know that this has this induced fit where oxaloacetate creates the acetyl-CoA binding site through conformational change. I do want you to know that part. What I'm talking about are the details of the enzymatic catalytic mechanism that involves the histidine. That is what I want you to know, and that there's a phosphoryl histidine, phosphohistidine intermediate. There's a succinyl phosphate intermediate. Those are the things I want you to know and how that works. So be able to talk about that. So here's where you get a nice little table that kind of outlines some key aspects. You get essentially these 8 reactions, and so we get citrate synthase, and a lot of times people will look at citrate going to cis-aconitase and then cis-aconitase going to isocitrate, because cis-aconitate is really that intermediate there. And so you can kind of think of it as 2 reactions catalyzed by the same enzyme. Has this prosthetic group, iron sulfur cluster, and then you have lipoic acid, TPP, FAD, very similar to the pyruvate dehydrogenase complex. And so even though I'm not asking you to know the specifics of that reaction mechanism, I am asking you to understand that it has a similarity to the pyruvate dehydrogenase complex, and I may ask you to compare the two in terms of just generalities. There's a lipoic acid, there's FAD involved in TPP. There's also NAD involved. Same things that are involved for the pyruvate dehydrogenase complex enzyme. Then you have, just other generalities, and I would like you to be able to recognize, in terms of the overall reaction, what's happening. And here's where we have condensation reactions happening. We have dehydrations happening. We have hydrations happening. Let's see. We have decarboxylations, oxidations, and substrate level phosphorylations. So I'd like you to be aware of what's going on there, to be able to recognize that something's being oxidized. I mean, this is a chemistry class, so I do expect that. Okay. Now let's talk about control. It's always a common thing to do whenever talking about a biochemical pathway. One of the reasons why enzymes are used to do these very energetic reactions, think about it. We could have just lit the glucose on fire and released the heat that way. Why not? It could happen. But obviously that would lead us to burn up, and that wouldn't be good. So what can happen then is, by breaking it up into enzymes, not only do we have the advantages that come with enzymes, in other words, we have the advantages that involve we can do these reactions under mild conditions where we don't need extremes in terms of temperatures and pH and that kind of thing. But we also, because we're using enzymes, have mechanisms to control those enzymes through allosteric control, and since they're proteins, through their production, the production of the protein itself, at the level of the protein or at the level of the transcript, which gets converted into the protein. So there's multiple ways that these enzymes get controlled. A lot of the time, the allosteric control is one of the major things that we talk about. But we covered that chapter on signal transduction, and signal transduction oftentimes can lead to a transcription control too, and that all is operative in these pathways. So let's look at the level of allosteric control. And one of the things I would like you to do is look at these steps and see if you can explain why it makes sense to have this level of control. For instance, why would pyruvate dehydrogenase complex be downregulated by ATP, by acetyl-CoA, and NADH? Why would it be upregulated by ADP and pyruvate? Why would that be the case? And then look at these. So the conversion from isocitrate to I said that the isocitrate to the alpha-ketoglutarate, where we get the oxidative decarboxylations. Why would that be a smart place to control? You know, and it kind of, decarboxylation reactions make sense to do this kind of control, because these decarboxylation reactions can lead to CO2 being released, and CO2, once it's released, is kind of irreversible steps. So those are good places to block, to control these steps at irreversible steps. That's one of the things you'll see as a common theme. Usually in metabolic pathways, the things that get controlled are rate limiting steps, are irreversible steps, or the first committed step. So like we oftentimes, when we get into different pathways, once a certain step happens, then that molecule is destined to go through that pathway. So those tend to be the steps that are under control by the cell, and that's certainly the case here. Once you get into these pathways, you can downregulate these steps. So why would NADH and ATP block these two steps? Why would that make sense? And why would ADP stimulate that step? So see if you can make sense of that. If you can make sense of that, why does it make sense? Then it's not so hard to remember. It seems random if you just try to remember these out of context. So rather than me go through it step by step, I actually am telling you, I want you to go through this and make sense of it. It has to do with, what these mean and how they're related to this cycle going through. I think you can figure that out. But the strategy of picking these steps, notice that's what they all have in common: decarboxylation reactions. And those are irreversible steps. Just a point I wanted to make with you. So I've already made I alluded to this next area, and that is when the TCA cycle isn't needed to extract energy from glucose, because it has so many high energy kind of molecules, that a lot of those molecules can become precursors to make other important biochemical intermediates. So this kind of shows you a nice little flow chart of how this can happen. So one is, first of all, so pyruvate, as we've been talking about, if we need the energy, then pyruvate will go through the pyruvate dehydrogenase complex, form acetyl-CoA, which will then go into the Krebs cycle. Well, sometimes all we need, are Krebs cycle intermediates, and we need oxaloacetate, for instance. One of the ways we can do that is through an enzyme called pyruvate carboxylase. And pyruvate carboxylase can add a CO2 to pyruvate at the expense of ATP, and since carboxylase, at the expense of ATP, it can add a CO2 to pyruvate. And that's, if you got lots of energy, then you can use ATP, to make the molecules that you need. Oxaloacetate can also be used to make through gluconeogenesis. It's a thing that we'll talk about during Dead Week. During the last thing we cover in this class. Oxaloacetate will be converted to glucose. And then oxaloacetate can also be used to make aspartate, and then aspartate goes on and it can be used to make other groups. Succinyl-CoA, we already talked about, can be used to make heme, other porphyrins. Chlorophyll also has a heme-like group, a porphyrin group. Alpha-ketoglutarate, well, if you aminate it, you can form glutamate. Other amino acids get made from glutamate, and you can also get to purines. Citrate can be used to make fatty acids, and it can be used to make sterols like cholesterol and that kind of thing. A lot of these things also come from the fatty acids, can also be made from acetyl-CoA, and acetyl-CoA, when you don't need it, can be used to store as fatty acids. It also can be used in the production of sterols, although where that happens is the acetyl-CoA actually has to be transported out of the mitochondria into the cytosol, where some of this chemistry happens, where you get the fatty acid synthesis and steroid production. So when you're using molecules for multiple purposes, and so we're using in the case we're making these molecules that are involved in the citric acid cycle. They need to be rapidly replenished because they're serving two purposes. They're used to make a lot of the molecules we actually need to live and survive, they also are harvesting energy as well. So we need to have replenishing reactions, anaplerotic reactions they're called, and one of them is that pyruvate carboxylase reaction I told you about. It helps make sure that we get what we need in terms of oxaloacetate. Oxaloacetate's used in the Krebs cycle, and it's obviously key to the Krebs cycle, but as I mentioned before, it's used to make other molecules, including glucose, through the process of gluconeogenesis. So that's when there's low ATP, we need to have ways of making sure that we get what we need in terms of the different precursors to keep up with both the needs of our energy needs, plus the fact that we also need to keep making these other molecules in order to have a healthy survival. So that's really that's where I'm going to end with this. The book goes into the glyoxylate pathway. I don't plan to cover that. So you have my permission to skip it, but if you want to learn it, you also have my permission to study it and get to know it as well. It's another pathway that helps plants and some bacteria live off of acetate rather than glucose. So anyway, there you have it. There's chapter 19.